Assalamu alaikum students, welcome to lecture 14 in biopharmaceutics, and we are discussing chapter protein binding. And in that, today's topic which we are going to discuss is types of binding proteins, and it is continuation of the previous lecture, which was introduction to protein binding. So in today's lecture, we are going to discuss about proteins involved in reversible protein binding, and we will be discussing particularly reversible protein binding because this sort of protein binding got a significance from pharmacokinetics point of view, and the other type, which is the irreversible protein binding, which we discussed, is the one which doesn't play a significant role in as far as the pharmacokinetics is concerned, but that is involved in toxicity. So the various proteins which we will get introduced to are albumins, alpha-1 acid glycoproteins, lipoproteins, and erythrocytes. So proteins involved in reversible binding: albumins, alpha-1 acid glycoproteins, lipoproteins, immunoglobulins, and erythrocytes are some important proteins which are involved in reversible binding. These are usually located all over the body, but mainly in blood. The highest concentration is found, and because of their highest concentration found in blood, these plays important role in maintenance of osmotic pressure, and apart from that, these also play important role in the transport of endogenous and exogenous substances. So first of all, we discuss about albumins. Albumins got a molecular weight of 65,000 to 69,000 daltons, and these are mainly synthesized within liver, just like other proteins. And uh, albumin is one of the main important plasma proteins which are involved in reversible drug binding to major extent. And these are distributed all over the body within blood and also in extracellular fluid of the skin, muscles, and various other tissues. Moreover, albumins usually constitute up to 60% of that in the plasma when they exist in the interstitial fluid, and because of that reason, albumins, by building up the osmotic pressure, play important role in maintenance of the water and so electrolyte balance within the whole body. So that is the main reason that during accidents, whenever there is extreme inflammatory conditions and capillary membranes are inflamed and wide open, the albumins usually go through the inflamed capillary membranes and get enter into the interstitial fluid. Now the concentration is above the 60%, which is the usual and normal, and because of this higher built up concentration of albumins within the interstitial fluids, what results is that a pathological condition arises which is called edema, or uh, overall the volume or the water content within the interstitial fluids will highly increase, and that will result a swollen body or edematic condition. Usually the albumin's level is maintained at a constant concentration of 3.5 to 5.5% weight per volume, or simply you can call it 4.5 mg per deciliter. And what to mean by weight per volume, that simply if you take 100 ml of blood, usually 3.5 to 5.5 grams of albumins would be present per 100 ml. This is how we measure the weight per volume percentage. So the important functions of albumins, as we just discussed, is the maintenance of osmotic pressure of blood, and um, moreover, these are also involved in the transport of various endogenous and exogenous substances. So the important endogenous substances which get bound to albumin and are transported throughout the body are albumins which make complexes with bilirubin. So let me tell you one thing here, that what is the significance when these endogenous substances get bound to be albumins or any other protein. Basically these are get transported to various areas within the body. Like if you remember, we already discussed biliary excretion, and bilirubin is the pigment, or you can say endogenous substance, which is excreted through the biliary excretion process. So in order to get reached to that place, albumins play important role by transporting bilirubin to that place of excretion. Similarly, it is the other endogenous substances like free fatty acids, various hormones like cortisone, aldosterone, and thyroxine. These are reached within the whole body with the help of these transporter proteins, and tryptophan and other compounds. So basically the important point to remember here and to distinguish between the transporting proteins and these binding proteins is that transporting proteins help to cross any drug through the biological membranes, while the binding proteins just play important role to reach that endogenous substance to that specific tissue or place, and later than that they don't play any significant role because these cannot help to cross the drug, that biological membrane. So here, this is something very important to distinguish between the transporter proteins and the binding proteins. So as far as exogenous substances or the drugs are concerned, as the lumens are basic in nature, so weakly acidic drugs, or which are also called the cationic drugs, get attracted to albumins. Examples are salicylates, phenylbutazone, and penicillins. Next protein is alpha-1 acid glycoprotein, and as the name is self-explanatory, it is acidic in nature, and that's why it will bound to basic drugs. Some important characteristics of alpha-1 acid glycoprotein: it is also called arosomucoid, and it is a globulin protein. Its molecular weight is 44,000 daltons and lesser than albumin's. Plasma concentration is 0.4 to 1%, again lesser than albumin. If you remember, in case of albumin's figure is 3.5 to 5.5%, which means that that figure is almost 3 to 5% higher than this one. So albumins exist in greater concentration. That is the main reason they are mainly involved in the binding processes, but those are involved with the acidic drugs because of their basic nature, while alpha-1 acid glycoprotein, because of its acidic nature, will mainly attract the basic drugs or the basic endogenous substances. So as far as the endogenous substances are concerned, globulins, or you can say the alpha-1 acid glycoproteins, have a low capacity. How you can understand the concept of low capacity but high affinity? As we just discussed that these proteins exist in a very low concentration compared to albumins. So because of the low concentration, their saturation or their chances of getting saturated are higher, and that what you call low capacity. And but higher affinity because of its certain structural characteristics and also its acidic nature. It has affinity for certain endogenous substances. Mainly those substances are corticosteroids. Among the exogenous substances or drugs which bound to alpha-1 acid glycoprotein, as we discussed, it mainly bounds to primarily basic drugs, and examples are propranolol, imipramine, lidocaine. These are the few examples. So lipoproteins are macromolecular complexes of lipids and proteins. These molecular complexes are um, a combination of lipids and proteins, and these are large molecular complexes. And based on the same factor, they might be divided into three classes because of having various densities, like VLDL, very low density lipid proteins, and LDL, low density lipids, and then HDL, high density lipoproteins, which are also called good cholesterol because it helps to remove the other cholesterols which are bad, which are actually very low density lipids, to remove from the bloodstream. So that's why these are considered good. But in fact, it's if its level is too high, that's also not good indication that might lead to atherosclerosis and various heart conditions, which are bad and definitely alarming. So, as far as endogenous substances are concerned, which are transported with the help of or which bind to lipoproteins, include plasma lipids, and few of the examples of plasma lipids are fatty acids, triglycerides, and likewise cholesterol. These are few important plasma lipids which are transported or which bind to lipoproteins and then transported within the body. And exogenous substances or drugs, mainly acidic drugs. And once the albumins get saturated, which is the basic protein, and which preferably bind to the acidic drugs. So if the albumins get saturated, the acidic drugs, if left over in the free concentrations, free unbound fraction, will shift towards the lipoproteins and will bind there. Now the erythrocytes, red blood cells, or the RBCs, which constitute 45% of the blood volume. And mostly about these proteins, we know that these are heme proteins, and these are oxygen carrying proteins. But they're also involved in binding with drugs and their transport. And as we know that blood is mainly composed of four major components: red blood cells, RBCs, white blood cells or WBCs, proteins, and water. And of course, water then is further composed of various electrolytes and endogenous, exogenous substances in trace amounts. So the drugs which are mainly got affinity towards RBCs or erythrocytes are acidic drugs, and these are phenytoin, phenobarbital, or amobarbital. These are the few examples. And however, this protein binding is basically a secondary protein binding, and it also depends upon the free drug concentration in the blood. Just in the case of lipoproteins, we discussed that whenever the albumins get saturated, the leftover drug, which is the unbound fraction of course, that gets attached to lipoproteins. Now again, in case of certain drugs which specifically got affinity towards RBCs, firstly they will get attached to the albumins. Once the albumins get saturated, the leftover would come towards and get bound to RBCs. However, if we look its impact on volume of distribution, so binding of drug to RBCs generally does not significantly affect the volume of distribution, because the drug is mostly bound to albumins in the plasma water. So a little portion you can say is bound there with the RBC, so that doesn't build a huge impact on the volume of distribution. So if we look at the impact of albumins on RBCs binding, like how these affect the extent of their impact on RBCs binding. So it is very interesting to know that phenytoin, which is an acidic drug, which got the affinity towards RBCs, apart from albumins. But if you look at the percentage which gets attached or bound to the phenytoin, and also that percentage which gets attached to the albumins, we see a huge difference. It is 25% of the drug which gets bound to RBCs, while 75% of the drug which gets bound to albumins. So the basic reason is the same, that this is the secondary sort of binding, and drug first levitate towards albumin because of the highest affinity towards albumin, and then after that, due to the secondary sort of binding to which the drug got towards RBCs, the drug will levitate towards this one as well. But as the percentage is small that is getting bound to the RBCs, so overall the impact of this kind of protein binding on the volume of distribution is not that remarkable. So, for such drugs, because of their great affinity towards RBCs and albumins together, the total whole blood drug concentration should be measured. As we know that usually whenever drug concentrations are measured, the samples which are separated or processed are usually plasma and serum samples. But in the case of drugs, like the example is of phenytoin, which got sufficient affinity towards RBCs as well. If we want to know the correct level of blood drug concentration, it would be better to measure the whole blood drug concentration rather than just plasma or serum. Okay, students, this was all for now, and in upcoming lecture, we will discuss about the impact of protein binding on volume of distribution and how it could possibly impact elimination of drugs as well. So basically we would be discussing about protein binding's impact on pharmacokinetics, and definitely the reversible protein binding we would be referring to, and secondly we would also discuss the clinical significance of protein binding in upcoming lecture.